We are this evening going to discuss animal eyes, and I'm often asked this question, what does my dog see? What does a fly see? Or a honeybee for that matter? And it's an interesting and perplexing question that has exercised the minds of many, many intelligent people in the field of optometry, vision science, and a lot of new research and data has come out. I'm going to go through some of that today. If I miss out your favourite animal, I apologise, because we could just do this on 3 or 4 different species of insects and be here for the rest of the afternoon quite easily. Here we see the beautiful eyes of this insect, made up of many, many facets, called a compound eye. Interestingly, there are other eyes present on this animal. It has 7, and I will tell you about those later on. If you're interested in this subject, there are a number of very good publications and overviews and reviews which are listed on this slide. In particular, a very good introduction would be Michael Land and Dan Eric Nielsen, and in fact any work at all that they've written I would recommend you to read. Now, there are many types of eyes. We've already seen that there are these compound eyes. We also see that even in the fossil record we have evidence of compound eyes. We have eyes that are camera eyes, and all these eyes look superficially the same, but in fact they're derived from completely different sources and they're completely different groups of animals. If you want to swim through a sticky fluid such as the sea, you want to make yourself shaped like a torpedo, and you need some stabilizing devices. In fact, you want to look a bit like a fish. And this thing that looks like a fish, in fact, is not a fish, it's a reptile. And this thing that looks a bit like a fish is, in fact, not a fish, and it's a mammal. And this thing that looks like a camera eye is, in fact, a camera eye, but it's from a cephalopod. This thing that looks like a camera eye with an iris that moves and changes is actually from a jellyfish. This is a camera eye, and this is actually from a true fish. And this is a camera eye that's from one of my colleagues. Now, this is called evolutionary convergence, and that's the independent origin of similar traits. And what it is telling us that there are only a few ways that you can build to move through sticky water, and this happens to be one of the best ones. And if you have it, you are likely to survive in that niche and not be eaten by something like this, and therefore you will pass on those fish-shaped genes onto the next progeny. It's not an accident that nearly all fish, certainly in the superficial zones of the sea, look pretty much the same. And that's the same story for eyes. Now, if we're going to design an eye, it's quite interesting to see the same evolutionary convergence in place. There are only really a set number of ways that you can design an eye. And since most animal species, the vast majority of animal species have eyes, that tells us that having eyes is really, really important. We take them for granted on a daily basis. But in fact, if we hadn't have had eyes, we would have been what dinosaurs euphemistically call lunch. And we wouldn't be here today. So there is an evolutionary arms race that goes on, and in the Cambrian period, when sophisticated eyes started to develop, they developed very quickly indeed, and it may well have been the invention of predation and predators that led to this rapid evolution of eyes. Eyes can be single-chambered, sometimes referred to as simple eyes, even though there's nothing simple about them, or they can be multi-chambered, or what we call compound eyes. And there are various different types of these. You could have a pit version, like a pinhole camera. You can have that in the compound version as well. You can have a refracting one, and you can use lenses to refract in various different ways. Apposing, appositional eyes. Above, superpositional. And there's various ways that you can do this. These are very interesting eyes, because unlike our camera eyes, these give you an erect image, not an inverted image. You can also reflect. And there's various different ways animals use reflections to get an image, and a formed image on their eyes. So that's it. Nearly all eyes look something like this cartoon. And that also is very interesting as well. Just very briefly, I'll just run through for the non-physicist in the audience what refraction is and what's happening with these systems with light. If we have a substance here that is more dense than a vacuum or whatever, air or water, as it comes through, the light rays slow down and then they speed up again. And if that is curved, they slow down and they skew and they can come to a focus somewhere over here eventually. These are the waves of light, and we imagine the waves of light and by looking at the troughs or the peaks, and they curve. And we can see this shape if we look at a harbour, watching ocean waves coming in. And light does exactly the same. And we trace that out using a ray because it's easier for us to think about rays than to think about waves. But essentially rays are just going at 90 degrees to the wave front. If we have a system where we have less dense material here, but dense material on here, let's say water, we can get refraction and we can get a focus. If it's very dense, we can actually get a focus with inside it. Otherwise, we get a focus here. And we do know from ancient times this was known. Pliny records that he read all the books of Rome through a bowl filled with water, which he used as reading glasses as he got older. If, however, the outside is the same as the inside, you lose this power of refraction and you don't end up with a very good focus. And this would happen, for example, with a pit eye in the ocean. And remember that eyes evolved in the ocean. So this isn't very good if you want to get really good focus. And if you want to get good focus, you've got to invent a lens that's going to focus these rays in water. Now here's the problem. We evolved in water with these eyes, then we come out of water, and bingo, we're back in this situation again, where we're in air and water. And this eye would be hopelessly overpowered for us. So we have to make adaptations as terrestrial animals. We make the lens a little bit thinner, we change the shape of the cornea, and ultimately we get the focus back onto the retina. Now, all optical systems are imperfect. If you remember as a child when you opened a cracker and you had those little binoculars or magnifying glasses and you held them up to the light, you saw coloured circles around the edge of them or very, very blurred images. Well, one of the blurred images is spherical lenses have spherical aberration and the light focuses at different planes. So if you have a very spherical lens, you're going to get more spherical aberrations. The other problem with them is you get chromatic aberrations, is that not all wavelengths of light behave in the same way. And red light is rather lazy and focuses far back here. Blue light focuses very aggressively. So we've got to deal with these problems because they are inherently part of the optical system. You've noticed here that with a large pupil or a large aperture, they are worse. So let's make the aperture small. Aha, we have a problem now. And the problem is when it gets small is this thing called diffraction. And you may remember from high school physics when you had to put powder on the water and then you put various things in here. Here come in the plane waves, and look what happens here. They spread out, and you get a bright, a dark, a bright, dark, and a bright. This is called an airy disk. And if we plot it out, if we have a large pupil or a medium-sized pupil, we don't get too bad a point focus. There's a bit of a spread. But as we make the pupil smaller, we get a bigger spread, and this gets worse. So we have conflicting problems here if we are going to design an eye. And the other thing we've got to do is that we've got to measure acuity, and it's no good putting the dog in glasses and say, read down the chart, because it's just going to go woof, and you say, Rex, is all it's going to hear too. So the Snellen fraction is not appropriate. So if we're looking at animals and measuring animal eyes, what we've got to do is to look at one good way would be to look at gratings. And these gratings can be either static in choice chambers, they can be rotating round, and we can look at the behaviour. I mean, you could imagine sort of having a goldfish in a bowl and rotating a grating round it and watch which way the goldfish swims, and various other things that can entertain scientists endlessly for years on end in between applying for grants. Now, if we're going to measure in cycles per degree, we can then talk about spatial frequency. So the frequency here is one cycle per degree, for example. Here, it's double, and here we have 2 cycles for every degree of visual angle. So 1, 2. This one only has 1. So we're going to talk about cycles per degree instead of Snell and Acuity, but I will convert it back to Snell and Acuity later in the talk so that we get back to kind of the anthropomorphic way of thinking about these things. And you remember that 1 minute of arc is subtended by the Snellen letter at 6 meters or 20 feet. So 2020 is just American for 6'6", because they still work in feet in these measurements. Now, how does that convert to cycles per degree? Well, we can have a look. This is the sine wave we've got here. We've measured this angle, then we come down. It turns out to be about, this is about 30 cycles per degree. So a human vision, you could say, is 30 cycles per degree, would be about 20/20 or 6/6 vision. We actually theoretically can see much better than that. But because of the optical aberrations I showed you before, we see somewhere between 30 and 60, which is the theoretical limit of the human eye. Now, when we're dealing with some animals, we can't even use cycles per degree. So what we've got to do is theoretically work out what they might see by looking at that retinal spacing. And this is called the acceptance angle. For an insect, we can look here at the spacing between the various receptors. For an animal, like a human, we can look at the spacing between individual receptors. And this is what happens. So a human can detect these bars at this distance, whereas an eagle, which has much closer packed receptors, can detect it much further away. And here we have a multifocal insect eye which can't see detail or spatial frequencies as well as camera eyes can, so they have to be a little bit closer to see the same images. Now, the cone spacing at the fovea is about 2.5 microns for a human, and this would give us this theoretical 60 cycles per degree. Now, using the theoretical acceptance angle, we can place animals, we can grade animals according to how well or what their acuity might be. Now, before I go much further, I want to tell you that acuity is not the most interesting thing about vision by a long way. It's the thing that stops you driving, but it's not what you use for driving, and that's the irony of it. But we'll come on to that later. So an eagle has terribly good vision. A human has reasonably good vision as far as the animal world is concerned, but most birds would think us pretty pathetic. An octopus is fairly good too. Cats, much worse than ours, probably a half or worse for acuity. Various gastropods, honeybees, more gastropods, bivalves, coming down, fruit flies, until we come down here to a lowly flatworm called planaria, which has one of the simplest eyes in evolution whatsoever. And you can see there is hundreds of fold differences between the visions of these various creatures. Now, apart from acuity, as we mentioned, we need to talk about contrast. Here is a high contrast target, and here is the same target with low contrast. And if it's big enough, you can still see it. But here's a high contrast target, and here's the same target in low contrast, and it's a damn sight more difficult to see. This is like driving in the fog. We're starting to lose the ability to see things. And this plots out, and it turns out that if things are very, very fine, we can't see them at this level of contrast. And if they're very, very coarse, we can't see them either, and there's this sweet spot which is defined by this contrast sensitivity curve. We can do that at a particular spatial frequency with this Pelly-Robinson chart, and this is used clinically, and you can see as we come down here, then eventually people stop being able to read all the letters here, and possibly only 1 or 2 letters on this line, and no letters as the contrast starts to fade into light grey into white. So that's contrast sensitivity. There are other things like movement and so on. I'll deal with those under the specific examples. The other thing that's interesting about animals is that most animals use a completely different vision system from ours that evolved 700 million years ago when vertebrates and invertebrate lines diverged. And what happened was that we use this way of packing our visual molecules, and this is called a cilia or a hair receptor. And if you want to remember this one, this is the one that looks like Marge Simpson. So she's got the big purple hair coming up, and that one's easy to remember. Whereas invertebrates, for their eyes, use this system, which are little villi. Now, villi are little things like you have in your intestine, for example, and they're little tiny finger projections, and they pack it in like this. So this is Bart Simpson. And what's interesting about these two different ways of packing, the biochemistry is different as well. We use a system activating this G protein, where we break up this circular molecule and open some channels with a second messenger that allows stuff to flood into the cell, changing the polarity, which then allows us to convert light into a chemical, then back into an electrical signal for transmission further down, an ionic electrical signal. Now, insects and other invertebrates, but insects in particular, use this system where the G protein, a different G protein, activates this enzyme that splits lipids. Lipids are fats in the membrane, so it damages the membrane. The membrane contracts, and as the membrane suddenly contracts, it physically opens the channels. So this is instantaneous. And this is why you can't swat a fly with your hands, because they can see faster than you can move. They can see much faster than we can. So what they're losing out in what we call spatial acuity, i.e. the number of bars you can see in a given distance, they're speeding up in temporal resolution. They've got much faster ways of seeing. And this is one of the reasons why. So, the other thing that a lot of animals can do, they can see polarized light. Now, polarized light's a bit freaky because we can't see it. So what we've got to do is imagine it. Light can vibrate in its waveform in many different directions, up, down, left, right, sideways, and a few others for measure as well. But if we pass this light through a polarized grating, we get rid of all these vibrations in all the other ways, and we only allow through light that's going in a particular angle, and that's called polarization. In fact, our atmosphere does this for us from the sun, and as light is scattered through the atmosphere, it becomes polarized, becomes linearly polarized, as it does if it's reflected off water, and you end up with this just vibrating in the same plane. And what's interesting about a lot of insects is that they use these Bart-Simpson or rhabdomeric receptors, and you can see they can act as gratings, and simply as the light going down here, they can detect polarized light in these different planes. It's a little more complicated than that, but essentially, if you can remember that, it'll be helpful when we talk about it later. Now, a little bit of Greek to deal with the pelagos, the open sea, and epipelagic means upon the sea, and this is the sunlit zone. And this is where most of the interesting stuff that we think about in the sea goes on. It's where all the food production occurs. You can have photosynthesis, you can have plants, you can have lots of animals, and you get jellyfish, tuna, sharks, and dolphins. They're all basking and enjoying themselves in these warm, superficial layers of the sea. Now, this layer becomes a little bit darky and murkier, and when we get down to 500 meters, about here, we get very depleted of oxygen, and therefore we need to develop things like efficient gills. We don't want to move much unless we have to. And here is established with swordfish and squid, wolffish, and some species of cuttlefish. And also we develop bioluminescence, that is, we start to make light ourselves, like zooplankton do. And if we can't do that because we're a vertebrate, we borrow them and we help them, and they live in with us, and therefore we can shine and see things. Then we go down to the bathypelagic, which is deep. Now this is a really strange, weird zone. Here is cold, it's totally silent. There is no light, there is no plant life. The only source of food is what's coming from above, foodfall, or other animals that are living in this zone as well. Marine snow. And here we have these gorgeous looking creatures, hatchfish, giant squid, some of the smaller squids, and some octopuses. And then we have the abyssopelagic zone, really at the bottom. This is very, very few creatures can live down here. Indeed, most of them don't bother with eyes because they're pointless, and they tend to be transparent. And the weight and pressure means there must be some body designs as well that can accept that. Let's go back up to those warm, sunlit epipelagic zones. You may remember this from a previous lecture. This is just a one-celled animal. It's one of those planktons that some whales like to eat, and ultimately dependent on Everything we eat from the fish comes from these animals. But some of them themselves are predators, and this is one. This one sits at the bottom of the water. It can sense light. It's got a focusing mechanism here. It's got a pigmentary layer here, which means it changes the colour of the light. And it's got a receptor here for light. And when it sees something coming above it, it leaps up and it eats it. And it's really quite astonishing. This is all in one cell, so it's not an eye. It's it's within the cell. And it's quite a complicated thing. When we develop the first eye that's truly an eye, it's got to have a cell, at least a cell. And funnily enough, there is an animal that has one cell that is an eye, one neural cell and one pigment cell to screen. And here it is. Why do we have that to screen? It needs to know whether the light is coming from above or below. And so if you put a pigment screen in, otherwise it would be firing all the time, it only fires when light is coming in from a particular direction where it's not screened. And you'll see its little finger-like projections in here which are packed. And what happens with this is it fires, the light comes in, it fires off, and it's connected to a contractile band here that starts to move, and it starts to beat its little hair-like things, and it turns, like the boats in Oxford and Cambridge boat race do, as you can see. And this rather elegant mechanism of responding to light and moving towards the light is really the basic level of animal vision. The next level is to have more cells packed into the pigment, and more pigment, and that would be a pigment cup eye that we see here in this very, very basic flatworm here. These are called lateral ocelli, or stomata, and there's various different types of these, and we see them in many, many types of animals, and they work like this. Because of the pigment, this cannot be seen unless it moves here where it can be seen, and suddenly it lights up, and the animal can then make a response to this shadow against the light or the light against the dark, depending on what it wants to do. Now, 85% of phylum of animals have eyes like this. They may have other eyes as well, but they have eyes like this. Some of them, this is the only eye. And those animals tend to have limited visual demands. They tend to live under leaves if they're on land, or they tend to live at the bottom of ponds. And all they have to do is to detect where the light is and move towards it, and detect if a shadow's coming over them, i.e. a predator, and move away quickly back into their burrow or under the stone or wherever it was they were hiding. So a very, very simple, basic eye, possibly from which all other eyes could have originated, as we showed in my last lecture on the evolution of the eye. Several receptors can actually occur within these pigment cups, and so we can actually end up with a significant response to relatively dim light compared to that single receptor that we saw earlier. Now, planarians are these small flatworms. They're rather cute. They've got eyes and they've got little ears, and these ears can detect the currents, and the eyes can see the light. And if you chop them in half, they regrow, and if you chop out their eyes, they regrow their eyes. So they're rather fun things for scientists who like doing that sort of thing to experiment on. And they found out some very interesting things, including some of these genes about regeneration and maintenance. And when we understand how these genes work, this could be very helpful for us, because it may help us, for example, cure macular degeneration if we're able to turn on genes that can make retinas from de novo, or from people who are born blind from congenital defects. It may be able to help them develop an eye. So lots of interesting work will come from this lowly animal. So they're not just of interest just of how they see, they're also of interest biologically, and they're also interesting to us, but not as interesting as these critters, which are delicious. And the main thing about molluscs is they have all different types of eyes, and many of them have eyes. Some of them quite complicated pit eyes and large eyes. Some of them have lensed eyes here. And why do they have all these eyes? Well, it's because what I said before, it's because they're delicious, and people like to eat them. And not just people, lots of other animals like to eat them as well. And they have very, very interesting eyes in that some of them can use these ciliary receptors, which is not what we expect with a arthropod. We expect them to use the rhabdomeric receptors. So there's things breaking the rules here, and we'll talk about that. Now, a pinhole camera, you will remember from your childhood, can actually make a sort of image on the back. And this is exactly what happens. The sort of image that can be made would be about 2.3 degrees when the pupil is small in this animal, but it can also enlarge its pupil dramatically to let in a lot more light. But now you can see, instead of stimulating just a few receptors, it's stimulating a lot of receptors, so the acuity goes down, and what it can differentiate has to be a lot bigger. And people have done experiments on Nautilus, and this is the one where they put it in a fish tank and spin round stripes of different sizes, and you can work out actually what the Nautilus can see. And it turns out the finest stripe has to be about 11 to 22 degrees, and that's the finest vision it can get. Now, why did the animal develop this vision to see rotating stripes? Because that doesn't happen in the real world. What it did was to keep it still and stop it rotating in its world. So stabilizing, what we call an optokinetic response. And we actually have an optokinetic response as well, which is when you spin yourselves round when you're a kid and then you stop and then you're sick and somebody looks in your eyes and you see your eyes doing this. This is related to this mechanism that is very, very, very old. Now, although it can see things, and it can see things fairly well through its pinhole pupil, it's very, very dim compared to a fish eye. An opening lets in more light, but it drops the acuity down, and it can't see very well. Does it care? No, because it lives in the deep ocean, and as I said before, this is living in very dim light, not much happens down there, not much moves, and what moves doesn't move very quickly. So they are very well adapted visually for where they live. I mean, they'd be hopeless trying to catch the tube or run around or do this lecture, for example. But where they live, they're fine, and they don't need to go any further and evolve a lens. Now, pinhole eyes also occur in these animals, which are giant clams. But they just didn't develop one pinhole, they developed a multiple in their outer mantle. So this is the really delicious edible bit, and then just here behind it is a little pinhole eye, another bit of delicious mantle, another eye, another bit, and they've got hundreds of these things arrayed around each of these cusps. And these are very, very, very good at detecting things coming towards them, because what they don't want is to have their mantle nibbled by passing fish. And a 10 centimetre fish at 42 centimetres will trigger the response. So it only has to be crude, but that's good enough, because those are the sort of fish that can do you a lot of harm. Now, they have these symbiotic algae that we mentioned before that live in the mantle tissue, so it's got to have access to sunlight to allow those algae to make the food that it needs to grow. So they don't want to close all the time, so they don't want an eye that's too sophisticated. If they had an eye that's going to close with every passing particle of dust, they would starve. So they need to have it open most of the time, and then only close it when a fish or another predator approaches. Now, take all those pit eyes and bring them all together in one place, and you've evolved a compound eye. And that's exactly what's happened with these arc clams. And here's the compound eye. Here's a compound eye of it, you can see. And it's essentially a lot of pit eyes brought together. It also has pit eyes as well, very much like the giant clam that we saw earlier. But this is the eye we're interested in now. And you'll notice that it has ciliated receptors, which is very unexpected for an arthropod eye. It doesn't have any lens, so it's got a very, very poor resolving power. Each of these holes may only have one or two of the receptors in, so it has a very, very large field of view, about 30 degrees, but that's enough for it to perceive spatial movement and trigger closure of the shell if a large shadow is detected. What's interesting about ciliated cells like ours, as we pointed out in my very, very first lecture, is that they are not interested in light. They're active in the dark, and light turns them off. So it's a very, very good defense mechanism for these animals to have. Now, we've said these pinhole eyes suffer because they cannot make a very good focused image, and things have to be relatively large to be seen. So here's the unfocused image that we're focused down here, or the eye would have to be incredibly large, implausibly large, to pick up an image from it, which is what the Nautilus does, and that has a 1 centimetre, an enormous eye for such a little animal in the shell. But again, still not big enough to get a true focus. So what we want is to do a lens. So we make a lens. And now look at this, we're focusing quite well. It's only 4 times the radius of this where the focus image is, and so we've got a nice little blur circle here, much better than this, and we can start to see things in much finer detail. And this is done, and here's a cross-section through a snail eye, gastropod snail eye, with one of these spherical lenses. And lenses don't necessarily have to be made out of congealments of protein. They can be made out of what's around in the environment, like aragonite. And aragonite is a very interesting transparent chemical crystal that has 2 refractive indices, and that's very handy if you spend your time in the intertidal shore like these West Indian fuzzy chitons do, because it means you can see underwater and you can see when the tide goes out. And again, it's the same thing. These things don't want to be eaten. They need to clamp down when a predator arose, and you can do visual experiments on them, and funnily enough, people have. And one they found out is that if they put a black disc 3 centimetres or larger, it clamps down. And that's about 9 to 12 degrees, which for us, a human eye, to make it so that we can do it, it'd be about 20 moons stacked up would be how big the thing would be. Now, a moon at arm's distance is half the sense of your fingernail, so it would be vision that would be seeing this, whereas we can see little, much finer than that, indeed. So it's pretty coarse. But it's sufficient for these things which have survived from the Cambrian period through today. And rather beautiful structures, as you can see. These are beautiful creatures as well. They're not flowers, they're worms. And they're very, very delicate tissue on them. And they live in the ground in these tubes that they make out of mucus and sand. Now, they've got to have eyes, and in fact they do, because they want to retract back into that tube as soon as anything comes by to have a little nose around and find out it's not a plant and it's an animal and eat it. And here are its compound eyes. What's interesting about these compound eyes is, 1, they're ciliated again, but 2, they're lensed. So they actually can get some sort of focus here, and this is going to improve their acuity. And they have about 12,000 of these onomatopoeias in each of these eyes. So they're rather large, and they're very, very good shadow detectors, because, as we've said, they're turned on by the dark, so they're turned on by shadow. So very, very good alarm devices, and Nielsen describes them as sophisticated burglar alarms. Now, the lenses that we are familiar with are within the eye, and here is an eye, and you can see the spherical lens virtually filling the entire eye, and we can see, because it's semi-transparent, the pigment layer even behind it in this slug. Now, different types of slugs and snails have their eyes in different places. Most of them have them on the end of these stalks, but some of them don't, and have them at the base of the stalks inside, particularly the air-breathing pond snails as well. So next time you go and see one of those digging out of your pond, have a look at it, and you'll notice that the eyes are not here. They're actually at the base of the tentacles. This fearsome-looking creature is a box jellyfish, and fearsome indeed. They are rather dangerous. Now, many jellyfish have developed pigment cup ocelli, the very simple eye that we mentioned before. But the box jellyfish has gone one further. In fact, it's got 24 further. It has 24 eyes, and it has 4 different types of eyes. And they're located in clusters, 4 clusters here. And this has within it 4 types of eyes. It's got slit eyes, it's got pit eyes, it's got 2 lensed eyes, one that always points up, because there's a little bit of stone in here that acts as a buoyancy to keep it on, and one that always points down. And these are camera eyes, and they can actually see spatial detail. And here is one of the eyes. This is the upper pointing eye. This is the lower pointing eye of the lens and iris. There's the pit, and there's the slit eye. And what sort of stuff could this see? Well, we look and see where it lives. It lives in the Caribbean mangrove forest, and this is the view that we would have exactly here. But if they go out to deep sea, they starve, because they're hunters. Unlike other jellyfish that float in and out on the tide passively, these things actually detect their prey and swim towards them to kill them and eat them. which is a rather scary thought, but luckily they're mostly small and they don't bother us unless we go for a swim, and they sting us, and their venom is so poisonous it can cause really tremendous damage to even humans. So here's a couple of these box jellyfish floating around here, and this might be what they'll see. The down-pointing eye is here to detect the mangrove roots, because it doesn't want to go and swim into one of those and bang its head, because it's rather, jelly-like and might cause some harm. And secondly, it needs to detect where the canopy is. It wants to swim here, and it doesn't want to go swimming off here and end up sort of off the shore of Norfolk or something. So it can detect the canopy, and that's roughly how it will see. It doesn't see in colour. It's got a single opsin as far as we know, and it detects coarse detail. But that's better than what it could detect just using its pit and slit eye. So then the question comes, why so many eyes? The reason is because they all do different things. This one is the eye for making sure that you go towards the canopy. This is the eye to make sure you don't bang into the canopy. This is the eye to tell you when it's daytime, and this is the eye that tells you when it's nighttime. And what happens, if you've got a lot of eyes, you don't need a lot of brain. If you've only got 2 eyes like us, you need a lot of brain and a lot of brain power. And since this animal is only a diploblast, not a triploblast, that means it's only got 2 layers in its germ cell. It can't develop things like hearts and muscles and brains and livers and all the stuff that 3-layered animals can. It's split off before the triploblastic line in evolution. So this is a very, very clever and sophisticated way of actually seeing the visual world. Here's another one. This is so cool, and it's beautiful. We only usually see the inside bit of this, and usually it comes with sort of black pudding and a bit of lettuce. But the bit we don't see is thrown away, and it's nice to go to a fish market and see it if you can, are these beautiful blue eyes, iridescent blue eyes. And they're there to actually stop animals going in and eating this. And if you think that in the United States in 2008 they harvested 53.5 million pounds of these things, you can see that it does need to do something about predators. Unfortunately, it can't do much about us, but it can do enough about the other ones to make sure there's enough left for us to eat. It has these little tentacles that filter feed and so on, and it doesn't want them nibbled by fish, so it's got to have this abduction response. It snaps shut at the approach, and the abductor muscle is the tasty bit, and it protects it. It's very good for you as well. It's low in fat, protein, vitamin B, selenium. There's lots of good reasons, if you're not a vegetarian, to eat scallops, and not too much mercury either. Now, how does it stop being eaten by other animals? Well, it's got a duplex retina. It's got a 2-layered retina. What's interesting about this retina, you see it's got the ciliated and rhabdomeric. Now, the problem is it's hopelessly underfocused, and light comes in through this lens, causing a large blur circle on these rhabdomeric receptors. So what's this for here? Well, very clever. It has a reflective mirror, and it reflects the light off, dead into focus on these ciliated cells. And we can actually make some estimates of what its visual acuity might be, and also on what its F number might be, which is the dimensionless ratio that tells us about the light gathering abilities of this here. These are the calculations which have been done in the Munich physics department, and we can see that it has an F number of about 0.5. And how we work this out, we know the focal distance, we know the diameter, we can divide the two, and we get a number, 0.5. Humans, for example, in dim light are 2.5, and in bright light, 8.5. Fish are 0.8, a good camera, 1.4. The lower the number, the better the light gatherer, and so what this is telling us, this is an extraordinarily efficient eye at detecting light. Okay? And it uses a very interesting mechanism, this argentea, this mirror, to do this. It's also got reasonable acuity of about 2 degrees. So focusing is critical if we want to see things in finer detail. But with a spherical lens, we know we can't really do that because the focal length is too long. But we know that fish can focus. So how do they focus with a spherical lens? And what it turns out is that they use a lens that is differently dense, different refractive indexes at different places in the lens, which means the light can now focus sharply onto their retina at about 2.5 times the diameter of the lens, which is all you need for a fish eye, which is excellent. And this is called Mattheson's ratio, from the person, or Mattheson's ratio, from the person who first did it over 100 years ago. Now, we can now get a sharp focus on the retina. And having a slightly flatter lens, we're going to get less aberrations, less spherical aberrations. And this type of lens evolves in cephalopods and fish. Cephalopods are octopuses and things like this. And here's some cephalopods for you. And look at their eyes. Beautiful camera eyes. These eyes look very superficially similar to our own vertebrate eye, but in fact they are derived completely different. They're derived from the epidermis, the skin, by infolding. Our eyes are evolved from an outpouching of the brain. And that's why our retina is inside out, and we have a blind spot, which these animals do not. So they look very much the same as our eyes, but in fact, they're very different indeed. And they have rhabdomeric receptors, and as I showed you in the second slide, this means they have the potential to see polarized light, and indeed they do. Squid, octopus and cuttlefish are masters of camouflage, and they use this outer layer of skin to disguise and change their colours very quickly with these chromophore cells. But what's interesting is the inner one. So we have multi-dimensionally rotating light that's non-polarized, which comes in, hits this iridophore cell, which is lined up very specifically, and this light then comes off, rather like coloured light off an oil slick. The thicker the oil slick, the redder the colour. The thinner the oil slick, the bluer the colour. And that's the same here. So the thin plates, beautiful iridescent blue light, deeper layers, the redder the light. Now what's interesting about this is that it comes off polarized, which is good, which is really interesting. And the other thing is you can block whatever colour you want with an appropriate chromophore cell. So you can now send a private signal that your predator can't. And the predator of this is a mammal. And that mammal is a sperm whale. So this animal can now signal to its friends in a private manner that sperm whales can't see. Polarized light's also used by mantis shrimps. Now mantis shrimps are the delicious food of the previous animal, the squid. So having polarized abilities here is not of much help, but it's got these sophisticated eyes that sit on the top here and move independently and look rather implausible. I mean, surely this is, just the wackiest bit of design that you've ever come across. And they're rather complicated, and there's several different layers in it, but we're interested in what's called the midband here, which also has layers, and we're interested in one part of the midband, which is here, and these are the cells that are specialized to see polarized light. But, as I've said before, so can your predator, so what's the point? Aha! What they can do is to see circularly polarized light. Not only is light can be polarized in a plano direction, it can also be polarized in a circular direction. And in fact, it can be circularized to the left and to the right. And these shrimps not only can detect that, they also produce it in their shells. And they're the only animals that can detect this, and it enables them to privately signal to each other from the safety of the reef without them being seen by their predators that can normally see normally polarized light. Fish eyes, very similar to ours, and that's not surprising because we evolved from some animal that looked like this some time ago. Unlike our eyes, they're more spherical, the lens, and the retinas have both, similar to us, rod and cone cells, but later in evolution we lost the ability of colour vision that lake fish and superficial sea fish have. They have much better colour vision than us. Some of them can even see into ultraviolet light. For example, juvenile brown trout see ultraviolet light reflected off the zooplankton that's their food. But when they mature and they become an adult and they're going to eat different things, they lose those codes. They don't need them anymore, and there's no point having them cluttering up your retina, and they don't see the UV light, which doesn't penetrate so well into the zone where they live, in the murky waters. Now, anchovies, fish, can detect polarized light. We're not sure why. Is it because of migration? Or is it because they can signal to each other when they're in a school swimming together. And that's actually thought a lot of schooling fish can actually have this ability to detect polarized light. Deeper dwelling fish, 1,000 meters, have very special adaptations because there's no light down here, or very little light. And here is a marine hatchet fish. And this is its environment. It's in the dark, with nearby an occasional flash of light from little tiny animals, these bioluminescent flashes. Now, most bioluminescence is blue, and there's a reason for that. It's because blue light penetrates through water and red light doesn't. As you know, when you go under the water, if you're swimming at all or snorkeling, the deeper you go down, the bluer things become. And if you haven't snorkeled or whatever, you can watch it on the telly with Jacques Cousteau, and that shows exactly the same problem. In fact, they have to adapt their cameras for colour underwater. Now, these are not degenerate. These are very sophisticated, and they have evolved in a world with almost no food, very, very low oxygen. These fishes are very weak, and they swim very, very slowly indeed. So they don't need to be able to see miles away. They only need to see as far as this animal, or maybe that animal is, just to get there. If they see further out, and they can't see further out here, there's no point, because they can never swim to it and get to it in time to eat it. So they have very, very good light detection, and some of them have extraordinary adaptations to their eyes. That actually is not the eye of this fish. That is a protective coating on the outside of the eye to stop the eyes being stung by the tentacles drifting down from jellyfish, and in particular from Portuguese men of war, which is why this is my favourite animal, because I hate Portuguese men of war, and I actually didn't know there was something that ate them. But there is. So although he's not the most handsome of chaps, he really does a rather useful function. It's a shame there's not more of them. Anyway, in this dark zone, we have to remember it's cold. There are no currents, nothing moves. It's almost completely dark. And you're going to evolve some very interesting things to live down here. One of them you might evolve is tubular eyes that point up, so that you can see the stuff coming down towards you against the very dim background reflection of the sky above you. Now, there are problems with that, because if you are swimming along and you've got one of these critters below you, you're going to cast a shadow, and it's going to see you, and it's going to slowly swim up and intercept. It's going to eat you. So you want to do something to make your underside light. So rather like aeroplanes are painted light blue underneath, so that they are counter-illuminated against the bright sky, so that their predators who are chasing them can't see them, or it's more difficult to see them. These animals do the same, and what they do is to use counter-illumination. They actually ingest, but don't eat, those little critters that make light, and they line them up on the outside of their body, and as they're up there, they now have the same illumination as the light above, and they don't cast a shadow, and they can be seen. Now, some of them do this in a rather clever way, and this is work from City University, as we pointed out before, from Professor Ron Douglas, who has done amazing work here and apparently spends his summer down in deep sea in submarines, where these animals live. Now, these are interesting because they release a chromophore that's red. Now, these animals do not have red receptors. No animals around there have red receptors, so they can't see this light. But one of these species can, because it has a red receptor, and the other one that makes the red light doesn't. But how it sees it is very clever. It actually ingests some animals with a chlorophyll pigment, and it absorbs the red light into the chlorophyll, which is green, and then signals from that chlorophyll to its blue receptors and green receptors, which now enable the animal to see in the red. So it can make light. It's like a private searchlight that no one else can see, and it can find its prey, and the prey doesn't even know it's being seen, so it doesn't swim away. And a very interesting way of doing it. We mentioned about mirrors, and funnily enough, there is an animal that uses a mirror to actually see in this eye, the downward pointing eye, part of its eye, and it also has an upward pointing eye. And this is a spookfish, brown snout spookfish, viewed from below. Absolutely incredible adaptation to living in the depth of the oceans. Now, you can develop very large eyes, like swordfish do, and very large eyes have advantages, as we mentioned before, more light gathering, and they used to remember swimming at deeper depths than most of the other fishes are. And it allows them to see far away. So it's 9 centimetres, about the size of a tennis ball. But what's the point of very, very large eyes? Because we're going to get more and more and more problems with aberrations. And eyes are expensive to build and maintain, so it doesn't make a point of making the eyes very big. So how do we explain the giant squid, and which has an eye the size of a basketball? Well, it evolved these eyes to escape its predator, the sperm whale. And it has a very, very large eye because it's sitting around in the depths, and it can see the flashes from the bioluminescence. What it does, though, is it doesn't eat those things. As the sperm whale dives, the bioluminescence scatters in the wake, and this can detect it and uses jet propulsion to squirt out of the way. Now, these huge eyes can pick up this tiny little speck of light from the other side of a rugby pitch. So very, very, very sensitive indeed. And they didn't just evolve as squids, they evolved in the past. This is an ichthyosaurus. This is Mary Anning, who in the 1780s collected fossils and sent them up to what was going to become the Natural History Museum. And they also had these colossal eyes as well. Why? Because they had these things, plesiosaurs, that were their equivalent to sperm whales that could dive deep. And they probably scattered light in the same way as the modern animals did, which is why they have the colossal eyes. Now, this is also used for detecting submarines, because they also scatter bioluminescence in the deep depths as well. Now, terrestrial compound eyes, we won't go into too much detail, except to once again mention Hooke, who was a previous Gresham professor in this college, and here is his drawing of the compound eye of the grey drone fly, whatever that was, probably a domestic fly. And they work in a slightly different way, as we've mentioned. They're compound, so there's lots of them. And remember, they're in this ice cream cone shape with all these little villi, with a light tube that comes down here to guide the light through to the sensing nerve and then out through the nerves to their brain to be processed. Now, that's a problem with that, is you're only getting individual bits, so you need to have a different form in the night time, and these are night insect eyes that actually pool the resources from different places and use sampling a much larger part of the world. So the night eyes are functioning very different, they're superpositional. And flies, even from an ancient time, use this neural superposition, which actually, instead of having them scattered over, what they do is pool the light from the various adjacent ice cream cones, and to give very good directional signals and improve their quality of light. Now, a part of spatial perception. Now, apart from these eyes, they also have these ocelli as well, and they've also got 2 eyes behind here, eyelets, which they're going to use for circadian rhythm. So, all in all, they have 7 eyes, the flies and wasps. We've mentioned their temporal vision is very quick because of this mechanism that physically opens this channel rather than the slow mechanism that we have that means that we've got to go through several channels. So they can also see in colour, and you can measure the colour vision of insects, and it turns out that they've actually got slightly different colour vision from us, and they can see into the UV, and by comparing this enables them to have a completely different colour spectrum, and this is needed for their lifestyle, like onion flies need to see little onions when they're in the blue-green, and also flowers and insect-pollinated flowers can do that. So what does an insect see? Well, this is the last thing a fly ever sees, according to Gary Larson. And it isn't, because it doesn't actually see like this, as I've shown you before, by the way it neurally pushes things together. Now, you can improve the acuity by making the eyes bigger, but unfortunately large insects like this no longer exist as the oxygen levels fell in the Permian era down to our current level of 20, so we'll never have large insects like this again. And it's estimated that this is what an insect eye, if we had insect eyes, that's what we would look like. Probably an underestimation, maybe bigger, and some people calculate they may even have to be the size of a house. This is what things look like with our eyes if we were to put things in ultraviolet, but remember they're going to pick up visual clues and colours and see things in a completely different way. This is the same flower, as it might appear to a bee, except for this wouldn't be red because it doesn't have a red receptor, and it's going to put all of them together and make a mosaic. And that's probably what a bee sees. Jumping spiders have evolved this particular beautiful way of using chromatic aberration to see. So they use the ultraviolet here, which is always in focus, this green, 4 retinas, which is out of focus, and by comparing how out of focus to the in-focus eyes, it knows exactly the distance you're away, and this critter leaps out and gets you very accurately. And it also scans the retina, as does this funny thing that lives in the Bay of Naples. Very tiny. It's got eyes here, it's got another lenslet here, and the lenslet and the retina move and scan the image. Very finally, we'll come on to the last group of animals, the birds, which have exceptional visual abilities in different ways because they pack in a lot of receptors. They've got relatively large eyes for their size, and to give us an idea of what it might do, a kestrel can see a 2mm insect from the top of an 18-metre tree, which is astonishing vision, really. Night ones have a tapetum. They have the reflecting one, as we're going to see in other animals that do night vision. Tremendous colour vision. They use oil droplets as well to enhance that. Their vision is probably 2010 or better, even 24. Much, much better than we can ever hope to achieve. Now, comparing us to horses, horses have quite good vision, dogs, 2075, and finally cats that have really poor spatial acuity. But what do we mean that they've got poor vision? Well, it surpasses, they're better than us for functioning in dim light because they've got this tapetum. They've got a rapid retinal response to another image, so they can respond very quickly to their eye symptoms. They've got better flicker fusion, they've got a better field of view, and they can differentiate many more shades of grey, and they can detect motion in their visual fields much better than we can. But they can't see colour very well. So Labradors, these seeing eye dogs, or guide dogs as we call them in our country, don't see the red and green, but they know where the red and green is. And so they function the position of where the brightness is. They also know about the speed of traffic. So this is what a dog might see with its colour vision, which is different from a colourblind human who has lots and lots of cones. Dogs don't have that many cones, so it's washed out all the way through, lots of shades of grey, and a neutral bit here where we're seeing greens. So when you buy Lassie, the little red ball, and you go and take it in the park, you're not doing Lassie any favours because it can't see it. It cannot differentiate this wonderful red ball against the green grass of your lawn. So when you have your dog and you're buying it a toy, think about what it might see, and it might actually enjoy something that may be blue-coloured or maybe yellow-coloured rather more than you might expect. And the final animal, of course, to not forget the cat lovers that might be here, also has a tapetum because although it has very poor spatial acuity compared to a human, it's not adapted to live like a human. This is a night predator, it's a night killer. And they are responsible for the loss of songbirds throughout gardens in Surbiton and beyond to this very day. So although they are cute and very nice and cuddly, you don't want to cuddle that. Otherwise, you're not going to be going out at night time again. And these animals are very, 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 very well attuned for the environment that they live in the wild. So, ladies and gentlemen, I haven't been able to include all animals, but hopefully the animals I have included, you've enjoyed having a look at how their eyes work. Hopefully it's helped you think a little bit about that fly and why you can't kill it. And also, understanding how these animals see is going to help us develop much more sophisticated ways for pilotless aeroplanes and cars and navigating around a complex environment. We may actually want to use something that's got a fast flicker fusion and maybe a compound eye rather than using camera eyes as we currently think we should be doing. So thank you very much indeed.